Good afternoon, students. In previous classes of antineoplastic agents, we were discussing about different causes of cancer, classification, and their mechanism of action. Then we were discussing about the different antimetabolites or methotrexate like this drugs. Now I'm coming to the second category, that is the antibiotics. How the antibiotics are working on anticancer as anticancer drug. First of all, we are coming to the drug that is known as the dactinomycin or actinomycin D. So it is a drug obtained from the cultures of Streptomyces antibioticus. It consists of tricyclic phenoxazine ring in the quinone oxidation state and two identical polypeptide. See, there is a L-methylvaline, and that is a series of amino acid, serine, proline, divaline, and L-threonine, where these polypeptides, where two polypeptides are joined together with two tricyclic phenoxazone. See, the cyclic rings are there, and it is containing phenyl and oxazone because oxygen and nitrogen is present in between the cycle. So mechanism of action, that is MOA, intercalation of the actinomycin into the DNA results in the inhibition of DNA and RNA synthesis. That is, the actinomycin drug will intercalate with the DNA, and as a result the DNA and RNA synthesis of the cancer cell will be prevented. Now coming to the anthracyclines or anthracinodions, that is also one category of antibiotics. So where doxorubicin and daunorubicin are two category of antibiotics, whereas we got the general structures. It is a four cyclic member where R1, R2, R3, R4, these are the substitute. In case of R1, R2 and R3, R4. In case of doxorubicin, all the groups are same, OCH3, H, OH and H, and in case of daunorubicin, you can find only for the R group hydrogen is there. So there is no more too much differentiation. So it works by blocking the function of topoisomerase 2, that is the one enzyme responsible for the DNA replication. Uses in leukemia and Kaposi sarcoma. Then coming to the bleomycin, that is having a very big structure of antibiotics. That's a complex antibiotics where different aminoglycoside and amino molecules are there and different amino acids are there. So bleomycin comprises of cytotoxic glycopeptides isolated from the strains of Streptomyces verticillarius. These are the glycopeptides, cytotoxic also. Bleomycin, a mechanism of action inside the cell. Bleomycin form a complex with ferrous, give rise to the hydroxyl radical and superoxide radicals. These radicals cleave the phosphodiesterase bond of DNA. That means this free radical क्या करता है, ब्रेक डाउन करता है फॉस्फोडाइस्टरेस बॉन्ड ऑफ डीएनए में जो फॉस्फोडाइस्टरेस बॉन्ड रहता है एक एडेनिन ग्वानिन ये जो प्यूरिमिडिन है उसके साथ. यूज़ कहाँ होता है, स्क्वामस सेल कैंसरस ऑफ द हेड, नेक एंड esophagus, skin and genitourinary tract. Now coming to the next category, that is the plant products. So that is the epipodophylotoxin, that is known as the etoposide. Whereas we getting the drug etoposide, this is a complex structure, aminoglycoside is present and a benzene containing OCH3, three rings are there. You have seen the structure as well as a plant from where we are getting this. It arrests the cells in G2 phase and causes of DNA breaks by affecting the DNA topoisomerase function, and resealing the DNA strand is prevented. That means ये DNA topoisomerase एंजाइम का साथ बाइंड करता है और उसका फंक्शन को बंद कर देता है, जैसे कि DNA तैयार होना बंद हो जाता है. Etoposide is a semi-synthetic derivative of podophylotoxin. दैट इज़ प्रोटोफाइलोटॉक्सिन से डेरिवेटिव मिलता है, उसको हम एटिपोसाइड सेमी सिंथेटिकली सिंथेसिस करते हैं. इट इज़ अब्सटेंड बाय द एक्सट्रैक्ट्स ऑफ मे एप्पल प्लांट, वन ऑफ द प्लांट. It is effective in the treatment of lung cancer, testicular cancer and Hodgkin disease. Now coming to the vinca alkaloids, that is the vincristine and vinblastine, that is derived from the vinca plants. Whereas in case of vincristine, R means that is a CHO, that is C in the N, that is one R group, and vinblastine R will be CH3. Now, mode of action coming to, it binds to microtubular protein tubulin and prevents the polymerization and assembly of microtubules and causes us mitotic spindle destruction. That means जो mitotic spindle बनता है उसको destruct कर देगा. As a result, cell का function complete नहीं होगा. The chromosome fail to move apart during mitosis and lead to metaphase arrest. इससे क्या होगा, क्रोमोसोम माइटोसिस में ये मूवमेंट नहीं कर पाएगा और मेटाफेज बंद हो जाएगा. Name properties and uses. Vinca alkaloid is isolated from the Catharanthus roseus, that is the natural plant. They have complex structure composed of a dimeric indole containing moiety named catharanthine and an indole containing moiety named vindolivin. See, we are just showing the structure again here. Two indole groups are there. So 4 closely related compounds have anti-tumor activity, that is the vincristine, vinblastine, vinrocidin and vinleuresin. Semisynthetic derivative of vinlorovin is also used as an anti-tumor agent. Now coming to the miscellaneous category, where cisplatin, it is containing platinum and amino group and chlorine. Cis-diamine dichloroplatinum is the name used in the testicular cancer. Metotane, that is one of the drug. 1,1-dichloro 2 chlorophenyl 2 parachloromethyl phenyl ethane. 2 phenyl group and 2 chlorine groups are there. CHCl2 and CH is present. Metotane, sold under the brand of Lysordin, is a steroid genosis inhibitor. That means steroid को inhibit करते हैं. And cytostatic antineoplastic medication which is used in the treatment of adrenocortical carcinoma. मतलब adrenal gland के कुछ carcinoma होंगे तो उसको prevent करता है. So thank you for watching this video, and just for more videos, watch, subscribe, like, share. Thank you.